So, welcome to this class on neuroscience of human movement. So, in this class we will discuss about animal preparations that are used in neuroscience experiments. In neuroscience, great knowledge in the last century has been obtained by the use of experimental animals or animal models to study movements, not just movements in other cases of in other neuroscience, right, but here we will only discuss cases that are used in movement related experiments, and the amount of knowledge that has been gained by these experiments is staggering. What are these various preparations that could be used? First is the case of spinal preparation, and there is a case where the transaction is happening above the level of the spinal cord. This is called as a decerebrate preparation. We discussed this briefly in the previous class, we will discuss it in some detail in today's class. And deafferented preparations, immobilized preparations, the other one that is popular these days is the neonatal rodent preparations, right. Neonatal means a baby. Okay. We will continue to discuss this. So, in spinal preparations, what is going on is there is a transection of the spinal cord at the lower thorax. So, what this will do? This means the hind limb or the legs. So, in quadrupeds, the hind limbs will be removed from the rest of the central nervous system. Okay. So, that means here is where that is happening. This is, so that means the nerves to the forelimbs are kept intact, whereas these are transected at that level. Okay. There are two cases, there are specific cases of two specific cases of spinal preparations. One is the acute spinal preparation, the other one is the chronic spinal preparation. The acute spinal preparation, the locomotor like activity is generated by the use of adrenergic drugs such as L-dopa and nialamide. So, when these are used, there is a spontaneous locomotor like movements that are present. So, in the acute spinal preparation, after the animal is surgically transected at that level, and after the recovery, this changes, as I mentioned in the previous class, this changes between animals. In some animals it can be as brief as 30 to 45 minutes, in some animals it can be several hours to several days. Okay. So, mostly what are the animals that are used in these experiments? Most of these in which we are interested in studying locomotion involve the use of cats. Okay. And it depending on the specific experiment, it can be others like rats and mice are in rare cases to study locomotion we do not use monkeys so much, but there are other cases in which monkeys are used. Okay. So, in the acute spinal preparation I said that spontaneous locomotor activity can be generated by the use of these adrenergic drugs, right. So, then there is one more case of spinal preparation that is called as the chronic spinal preparation. These animals actually recover activity after some time, and the performance is not going to be normal. Of course, you cannot expect that the walking is going to be normal after the surgery, but it will be not so much different, or in other words, in the sense that the function or much of the function is still retained, one would be surprised that wow, after this animal is able to do that. This does not happen in humans, let us remember that this does not happen in humans, or at least it takes several weeks for this to for any semblance of the function to return in humans, but not in animals. In animals I said that there is fundamental difference, right, we discussed this in the previous class. Okay. So, check the case of chronic spinal preparation, please Google that. Okay. Then there is the case of decerebrate preparation, invention or discovery of Professor Dr. Charles Sherrington. Right. So, Sherrington came up with an interesting approach to studying cat locomotion. What he said was that okay, if we could transect at the level of the midbrain, right. Then what happens is, if the cut, if the surgical cut from the higher centers is made at the level of the midbrain, then while keeping the midbrain locomotor centers intact, what happens is there is great extensor tone that is maintained, and this great extensor tone is sufficient to support the body weight of the animal, right. We call this, we said this earlier, and we call this as the decerebrate rigidity. In previous classes we have described this. Okay. So, this was the first description by Charles Shenton, where in future this model was refined, and there it came to be, there are at least now 2 types of these preparations. These are pre-mammillary preparations and post-mammillary preparations. This is the case depending on whether there are small bodies in the brain called as mammillary bodies, that is not relevant to motor control. So, they will not be discussed here. So, depending on whether the transduction is made before or after the mammillary body level, these two are called as pre-mammillary preparation and the post-mammillary preparation. Okay. So, and this is also called as the mesencephalic preparation. Okay. So, mesencephalic preparation means midbrain preparation. So, okay. So, depending on which one is used, the responses will vary. If it is the pre-mammillary case, right, then the locomotor regions, there is going to be spontaneous locomotor activity that is going to be seen. But in the other case, you need to have electrical activation of the midbrain locomotor region. So, if I appropriately stimulate the midbrain locomotor regions, I am going to see locomotor like activities. But in both cases, if the cat is made to walk on a treadmill, so here is a treadmill, you are wondering what this is actually a treadmill, and the cat is walking on it. It turns out that in both cases, if the cat is made to walk on the treadmill and I change the treadmill speed, the cat's walking speed changes in accordance with the treadmill speed, something that you would expect. So, unbelievably, let us remember what this case is. This is the case of transduction at the level of midbrain, or the control or the descending inputs from the cortical region or the cerebral cortex, especially the motor cortex is removed completely, right. But what we are doing is we are retaining the influence of the locomotor regions within the midbrain. So, in the midbrain there are circuits that are responsible for locomotion. So, these are kept intact. So, and when we do these experiments, it is found that these cats can walk according to the speed of the treadmill, right. And again there are 2 specific cases, one is the mesencephalic cat or the post-mammillary case, and the other one is the pre-mammillary case. Okay. Again many of these are used to study reflexes, to study central pattern generators, etc., right, these preparations. Now, another case is the case of deafferentation. Very controversial procedure. This is not popular these days, less used these days, but in the good old days, what used to be the idea is that long time ago, more than 100 years ago, the idea was that one inputs or sensory inputs due to one movement that is made. So, when I am taking my leg and keeping it on the floor on the next step, then I know that I have kept the leg on the floor. So, that is due to the sensory information. That sensory information triggers the next reflex of the other leg, for example. So, the next movement is a reflex that is caused due to the sensory input from the previous movement, and the next movement is going to be caused by the sensory inputs is basically a reflex that is caused due to the sensory inputs of the current movement and so on and so forth, right. How this is happening was not clear at that time. In the good old days, this was believed, and so one way to affect or control or reduce or completely remove the sensory information is to transect or is to cut the dorsal root of the spinal cord. So, the cut is performed here. Let us remember what does the dorsal root do. We have discussed this in previous classes. The dorsal root receives only afferent inputs. All the receptors, muscle spindles, Golgi tendon organs, cutaneous receptors, everything come via the dorsal root. If I cut it, no sensory input is going to be there, right. Then what is coming is purely not due to reflexes, is purely due to sources other than reflexes, is it not? Ventral root is responsible for motor, we know this is motor or efferent. The dorsal root is sensory or afferent, is it not? So, we have seen this in previous classes. So, when I cut the dorsal root, all the sensory input are removed. So, that means reflexes cannot be elicited, there can be no more reflexes in these cases. So, what happens is due to other sources. So, this was a relatively popular model or animal model some few tens of years ago, several tens of years ago, but not anymore. Nowadays this is not used. A particular case of deafferentation was used in the development of or training the animal to use an affected limb, right. So, this became a very controversial case. I have not described the case here. So, those who are interested can check the silver spring monkey case. What happened here was that the monkeys were deafferented on one side of the body. So, this is an approximately so that means they will not be able to sense that side of the body, but the other side was kept intact in that case, right. So, and so the other side is the good side, right. If I am restraining the good side from performing any motor action, so let us say that the right hand is not able to sense, but can perform motor action. The left hand can sense and perform motor action, but it is restrained from performing motor action. Then if I have to do any activity, let us say there is a piece of food that I want to pick up, I cannot do it with the left hand because it is restrained. I will then be forced to use the limb that does not have sensory inputs. Does it lead to plasticity? How does it lead to plasticity was the controversial experiment that was performed in the 1970s and 1980s in the lab in Silver Spring, right. So, you could check the there are several problems that come with deafferentation, absolutely no sensory information, right. So, that means if this part of the body is hurt or injured or pain cannot be felt, right. So, pain is, by the way, is useful for us to address a problem. If there is no pain felt, it is a huge problem, it is a bigger problem than pain itself. So, because we could cut ourselves, but not realize that we have cut ourselves, dangerous, and it could lead to more serious complications, infections and so on and so forth. So, do check this, Google this Silver Spring monkey case, you will know about this. I am not presenting it here for other reasons. So, okay. Then the case of immobilized preparations, in these cases what happens is that the animal is immobilized with the use of tubercularine. We said what this does, right. Early on we have studied this in the case of neuromuscular junction long time ago. When we discussed the neuromuscular junction case, we said what are the various things that could go wrong. At that time we described tubercularine. Remember, tubercularine is used as an arrow poison in the Amazonian rainforest, etc., and is an inhibitor of acetylcholine by competing with acetylcholine for the receptors, for the ligand gated channels or the nicotinic cholinergic receptors. So, it basically takes over the nicotinic cholinergic receptors, blocking the reception of acetylcholine at the neuromuscular junction. We said this is the mechanism of how tubercularine functions, right. In these cases, so when tubercularine is administered, neuromuscular junction does not function. So, the neuromuscular junction cannot function and cause actual muscle contraction. If actual muscle contraction is not happening, that means movement will not happen. If movement does not happen, proprioceptive inputs are, if only when a movement is happening, stretching or force production can even be detected by the sensors, muscle spindles, Golgi tendon organs, etc. If there is no movement or if there is immobilization, right, if that is happening, then afferents will not be firing, there will be absolutely no proprioceptive inputs, is it not. In that case, if I can selectively record from alpha motor neurons that fire specific muscles, you will have locomotor like activity in the alpha motor neurons or no real locomotion. Real locomotion involves actual movement. Locomotor like activity in the alpha motor neurons that control the locomotor muscles. So, if the recording is in the alpha motor neurons, you have a case that is not real locomotion, but fictive locomotion, right. So, this is called as fictive locomotion, do check this. Okay. So, in this case proprioceptive reflexes is removed, whereas tonic sensory input is preserved. So, this is a special case of immobilized preparations. Okay. Then there is the more recent preparation of the neonatal rodent preparation. In this case, from a neonatal rodent, so it is a baby, it is a pup, right, between 0 and 5 days. So, the spinal cord is removed and kept in a saline bath, right. So, and it will generate coordinated active bursts of activity in the motor neurons, in the locomotor like activity in the motor neurons when exposed to specific chemicals. What are these? Serotonin and NMDA. When these are exposed, so you are going to see locomotor like activity or bursts of activity in the leg motor neurons. If these are used to identify which neurons are responsible for making which movements, etc. So, to identify circuitry within the spinal cord or the spinal circuitry, if I want to identify, this is a very useful model. Okay. So, what we have seen in this class so far is spinal preparation. This is the case where the spinal cord is transected at the level of the thorax. So, this is the spinal preparation, this is what we have seen. Then we have said we have seen the decerebrate preparation. There are two cases of this. This is a pre-mammillary preparation or the post-mammillary preparation of the mesencephalic preparation, right. And then deafferented case, the case in which the sensory input is completely removed by cutting dorsal root. By the way, let us remember, it could be one side, it could be unilateral or bilateral. Deafferentation can be done on one side or on both sides. Okay. The silver spoon case is the case where it is done unilaterally, check this anyway. Okay. It need not be done on both sides. And immobilized preparations, immobilization is induced through tubercularine, which competes with acetylcholine and blocks the function of neuromuscular junction, right. So, removing the proprioceptive inputs and causing a sort of locomotion that we call as fictive locomotion. Then neonatal rodent preparation, in which the spinal cord of a baby rat is kept in a saline bath. Okay. So, these are the various preparations that are used in various cases. And what is the knowledge? Why do this? The knowledge that is gained from this has been used to improve human health in so many different ways. Much of the knowledge about and several about the movement related function and in other cases have been arrived at by studying animals, and rehab protocols have been developed by studying animals. However, a question remains as to what is the right thing to do. is it right for us to use animals to improve human health? That is an ethical question, that is a philosophical question, that is definitely pervasive. At least the following are recommended. There exists what are called as animal ethics committees, institutional animal ethics committees that control what kind of protocols are acceptable and what are not acceptable. So, and which animals. So, depending on the animal, specific protocols are permitted and specific protocols are not permitted. Experiments that are performed on animals will have to go through an institutional, usually will have to go through an institutional animal ethics committee that controls which experiments are accepted on which particular species and which are unacceptable, and what are the things that you should do, like whether we should what kind of maintenance should be provided to the animals, whether food should be provided, whether at what times, how they should be maintained, and whether veterinary care should be provided. All these things are controlled by the institutional animal ethics committee and that enforces basic things for the existence of these animals. Some of these animals continue to live for a long time after going through the injuries. These let us remember, these injuries are induced surgically by the humans, by us, by the experimenters, by the researchers, right. They continue to live for in some cases for years, right. So, that means maintaining animals that are no longer suitable for experimentation, that also becomes a question, like people will be willing to maintain the animal in the animal house for as long as I perform the experiment. What after that? Suppose the animal is too old, should we simply sacrifice it, or should we do something else? What are the options? All these things continue to be questions of ethics. Debates happen between animal rights groups and researchers all the time, but at least one thing is clear, that animal research is here to stay, and animal research has contributed greatly to the improvement of knowledge in neuroscience. So, with this we come to the end of this class. So, thank you very much.